Hi guys. Okay, so today I'm going to be walking you through the steps of muscle contraction and sliding filament theory. So I'm going to start by just reviewing what we already know about the hierarchical structure of a muscle cell. Muscle cells we represented with spaghetti when we were modeling a muscle, and then when we brought lots of muscle cells together to create a fascicle, we wrapped it in a membrane called the perimyocin. Multiple fascicles come together and are wrapped by another membrane called the epimyocin, and that epimyocin comes together to join to a bone as the tendon, so that the muscle can pull on the bone and move those joints. So we're going to zoom in on just the muscle cell, this fiber right here, so that one piece of spaghetti. So muscle cells are made up of even smaller units, so that's not the smallest unit. You could go down even further. So they're composed of things called myofibrils, and myofibrils are these stripy, small tubes. Muscle cells also have multiple nuclei, so that's what these bumps are on the outside. So the fiber is multinucleated. We're looking at the stripy pattern now. So the reason that the myofibrils have these horizontal stripes on them is because they're composed of two different kinds of filaments. So those are the actin filaments and the myosin filaments. Actin filaments are these thin blue lines, and the myosin filaments are these thicker red lines. So actin is longer than myosin, but myosin is thicker bands than the actin is. We need to pay very close attention to how we're going to label these different parts of the myofibril. The first thing I want to draw your attention to is what's called the sarcomere. So the sarcomere is going to span from one Z disc to another Z disc. And this is the thing that actually is able to shorten. So these blue lines, these actin filaments, will come closer together. Those Z discs will come closer together when contraction is occurring. The I band is the region of the myofibril that only contains actin. The A band is the entire region that contains myosin. So the entire length of the myofibril that contains myosin is called the A band. The H zone is the region located in between the actin filaments that only contains myosin. So the H zone is what's actually going to become shorter when that contraction is occurring, because those actin filaments are coming closer together, those Z discs are coming closer together, but the myosin does not change. Those thick filaments do not change in length. So taking a little bit of a closer look at that, we can see again the H zone is the region where only the thick filaments are located. The A band includes all of the myosin. The Z line is the location where a sarcomere would meet the actin of another sarcomere. And the I band is the region that only includes actin. So that goes between two different sarcomeres because we're crossing the Z line here. The reason why the myosin has these little nubs on it is because it actually is able to bind to these actin filaments. So they're called myosin heads, and that's what's going to bind to the actin to pull it along the length of the myosin and shorten that myofibril, that contractile unit. When we look at this under a microscope, we're going to see that pattern again. So the Z line, the region where those two actin filaments from two different sarcomeres meet, is those very dark lines under the microscope. This region here is the I band that contains only actin. These regions here are where the thick filaments overlap with the actin, and then in here this H zone is where only the thick filaments are located. So the H zone is what's going to shorten because the actin of the two different sides of that sarcomere are going to come closer together when contraction is occurring. If we look a little bit closer at those actin filaments, we're going to see that they're not composed only of actin. So down here, you're going to see these greenish blue balls. Those represent the actin. And they're intertwined with what's called tropomyosin. So tropomyosin is this pink rope-like structure. And then that tropomyosin is then bound to a molecule, these white balls here that represent troponin. So troponin is a molecule that binds to tropomyosin, and tropomyosin wraps around the actin filament. That's going to become really important later when we start talking about how your muscles stay under voluntary control and how we prevent them from contracting when we don't want them to. The myosin is the thick filament, like we said. It's made up of a lot of these smaller rope-like structures, and then the heads are on the ends of those. So these heads are the things that are going to bind to the actin when we send a nerve signal for them to do so. So just to put that all together in one diagram, we can see the myosin down below, the myosin heads here. Our actin is this pink structure in the middle, and it's wrapped up in tropomyosin. Troponin are the molecules that are attached to tropomyosin. The reason why tropomyosin and troponin are important are because the actin has binding sites for these myosin heads, but they are covered up by the tropomyosin until calcium is able to bind to the troponin, move that rope out of the way, and make the binding sites available for the myosin heads to bind to actin. So until calcium is available and until it binds to troponin, those binding sites are completely covered by that tropomyosin rope. So we need calcium to make them available for myosin binding. So that brings us to sliding filament theory. This is what you're going to be modeling and sharing with the class next week. The first step of sliding filament theory is that ATP needs to be available for the myosin heads to change position. So they're going to lean backwards in what's called the high energy configuration when ATP is hydrolyzed into ADP plus pi, because that releases energy and allows those myosin heads to change positions. The second step is that those high energy heads are going to form a bond with the actin complex. That bond is called a cross bridge. So when the myosin heads are able to bind with the binding sites on the actin, they form a cross bridge. After that, myosin relaxes into the low energy configuration, so it's changing shape again. And when that happens, that's what pulls the actin filament along the myosin, shortening that H band in the middle. So that pulls the actin filament towards the center of the sarcomere. And that cross bridge is going to stay formed even after the movement has occurred until more ATP is made available to break that cross bridge. So that's going to become really important when we start talking about rigor mortis. So this is a cartoon of that entire process happening. Step one is showing your myosin head here, and that is, let me expand this a bit. Here we go. Okay. The myosin head here, and that's in the relaxed position. When ATP is made available, then we can shift to our high energy configuration. When the tropomyosin, that rope, has been moved out of the way because calcium has bound to troponin and pulled the tropomyosin off of the binding sites, at that point our cross bridge can form. So that's what's shown in step 3 and 4, is that cross bridge formation with the myosin head between the actin. After that, we do the power stroke step. So that's when we switch back to our low energy configuration and pull the actin along the myosin filament. That cross bridge is going to stay connected until more ATP is made available to release the actin from the myosin filament. So I'm going to show a quick animation here that goes through that entire sliding filament theory.
We move our bodies by contracting skeletal muscle. We move our bodies by contracting skeletal muscle. While the results of this action are plain to see, the mechanism behind the action is not. Contraction of skeletal muscle tissue occurs when actin and myosin myofilaments slide past one another, causing the sarcomere to shorten. It is important to note that the individual myofilaments do not change length themselves, but instead overlap each other to shorten the sarcomere. Moving in for a closer look, we can see the details of how this occurs. The molecules you see here are myosin, which at rest has both adenine diphosphate and phosphate molecules attached at the individual myosin heads, actin, tropomyosin, and troponin. When we decide to move, calcium is released from the sarcoplasmic reticulum. These molecules bind with their troponin molecules, This causes the tropomyosin molecules to move, revealing myosin attachment sites on the actin myofilaments. The single phosphate attached to the myosin heads gets released as the heads move to connect with the attachment sites on the actin. The resulting formation is called a cross-bridge. The remaining adenine diphosphate molecule is then expended in what is called a power stroke, when the myosin heads pull the actin inward. Once the adenine diphosphate attachment is spent, adenine triphosphate molecules attach themselves to the myosin heads. These molecules trigger the release of the myosin heads from the actin attachment sites and are immediately broken down into adenine diphosphate and a single phosphate as the heads move back into their resting position. This is called the recovery stroke. If calcium is still present, the cycle will repeat again, and the sarcomere will continue to shorten until the calcium ions are transported back into the sarcoplasmic reticulum and the muscle relaxes.
As far as the role of tropomyosin and troponin and how that sliding filament theory operates. So the next thing we need to talk about is how that whole process is regulated, because we only want our muscles to contract when we send a nerve signal to them to tell them to contract. So we need to keep all of our skeletal muscle under voluntary control. The way that's going to work is by taking advantage of the fact that that tropomyosin requires calcium to be removed. So it's only going to happen, the calcium is only going to be made available when the muscle cell gets a signal from a motor neuron, and that signals the release of calcium. So again, calcium is going to be binding to those smaller molecules, those troponin. When that happens, the troponin and calcium complex is able to pull the tropomyosin away from the binding sites where the myosin heads needs to attach. So all of that calcium is coming from the sarcoplasmic reticulum, which is sort of a mesh network that intertwines all around all of those small myofibrils inside of a muscle cell. So we're going to take a closer look at that. Here's a representation of that entire process of where the tropomyosin is pulled away. So these tiny little dots here in the top diagram are covered by that rope, that tropomyosin rope. Troponin is attached to that tropomyosin rope, and when calcium ions are made available to bind to the troponin pulls the tropomyosin out of the way, and then those myosin binding sites are exposed and they're able to bind to myosin heads, so that actin can then be pulled along the sarcomere. So up here, you're going to see the sarcoplasmic reticulum, that sort of mesh network. This is what stores all of that calcium. So the calcium is actively transported up into the sarcoplasmic reticulum, which means it requires ATP. So that's how we get that very high concentration. We work against that concentration gradient to store a ton of calcium in this mesh network here. The sarcoplasm is just like the cytoplasm of another cell. We call it the sarcoplasm when we're talking about the muscle cell. And the sarcolemma is the membrane. So this is what's going to receive the signal from your motor neuron. The other thing that's important to notice here are the T-tubules. So that's part of the sarcolemma, part of that membrane that extends down into those myofibrils so that it can make contact with both the sarcoplasmic reticulum and those myofibrils. So that's going to become important when we start seeing how that action potential actually causes the release of those calcium ions that are stored in that sarcoplasmic reticulum. Okay, so this is a textbook image of that entire process. So up here, we have the terminal of our axon. This is a motor neuron. So if you remember how action potential works, the action potential is propagating down this axon until it reaches the axon terminal. At that point, we have neurotransmitters stored in the axon terminal. In this case, we're talking about muscle contraction, so the neurotransmitter we need to use is called acetylcholine. So that's the ACH here, these little red boxes. That acetylcholine is released into the neuromuscular junction. So when that happens, the acetylcholine is able to bind to the sarcolemma, that membrane that surrounds all of those myofibrils. And that happens at the motor endplate. So that's going to trigger another action potential along the sarcolemma. So those acetylcholine molecules bind. The action potential is going to travel along that membrane, along that sarcolemma in each direction. and then it goes down into a T tubule. So that's what goes through all of the sarcolemma and down deep into those myofibrils. When the action potential travels, it's going to signal another type of receptor called a DHP receptor. So this is a voltage-gated receptor. And it's coupled with another receptor on that netting, on that sarcoplasmic reticulum. So this one is mechanically gated, and when that DHP receptor opens, it also causes the receptor on the sarcoplasmic reticulum to open, and that calcium, that very high concentration of calcium, floods out, which makes it available to bind with troponin, and that removes the tropomyosin out of the way of the binding site so that the myosin heads can attach to the actin. So this is another representation of the same thing. I'm not going to go through it again, but you can certainly pause the video here and take a look if you think it will help you to see it represented in another way. Our T tubules, acetylcholine is happening here, and then you can see how that triggers the release of calcium from the sarcoplasmic reticulum. This is showing the entire axon here, so if you remember where the nodes of Ranvier were, that's where the action potential is jumping over the myelin sheaths. When the action potential reaches the axon terminal, that triggers the release of these little red dots, these acetylcholine molecules, which can then bind to the motor end plate and trigger the release of calcium ultimately from the sarcoplasmic reticulum after it travels down the T tubule. Okay. So next week what we're going to do is we're going to be doing a lab to test the effects of calcium and of ATP on muscle fiber length. And you're also going to be sharing the videos of your entire model of the sliding filament theory. So the rubric is posted on Google Classroom. Make sure you use that during your videos. I want the parts to be moving, and I need you to include several of those key terms that we just talked about in today's video. So if you have questions, please don't hesitate to email me. This is confusing. This is a lot of content, so try not to get frustrated with it. You're going to need to watch this video several times, and I'm going to provide some more resources as well.